Good morning, all the guests, and the Dean of the Faculty is also in this room, which made me very happy. When the seminar first announced, it seemed like there would be only 5 persons to attend this seminar, this special lecture. When we send slides and post on Facebook, there are many people, inform me that they would like to come. But behind that, I know some of you are graduate students, and if you attend the seminars, you get some special points. That's what I heard. Anyway, it's very good. I will start with report to the dean, and then the dean will deliver some welcome remarks and opening remarks, and after that, Dr. Thanrat Kaepajang will tell you the biodata of Dr. Alexander Uben, and the seminar will start very soon. This seminar has been arranged and now is going to happen because of Dr. Alexander Uben had the fellowship from Asia Uninet to do the short research in Thailand for 2 months, started in July, and it will be end tomorrow. He and his family will go back to Austria tomorrow. The research project that Dr. Alexander proposed to Asia Uninet and was approved by National Research Council of Thailand is that diversity and trophic roles of Macrofungi in different forest types of Sakaerat Biosphere reserve. This research, Dr. Alexander is the leader. Mr. Thaksin Achawakong, Dr. Thanrat Kaepajang, and myself are co-researchers. It's only for 2 months, and I heard that Dr. Alexander Uben found many mushrooms in Sakaerat. We have 2 big groups of people from Department of Microbiology. Raise your hand, please. Who came from Department of Microbiology, led by Associate Professor Dr. Vichawanit. He's my classmate. The second big group is from Department of Plant Pathology. Raise your hand. Okay, this is the department that I graduated from. I studied bachelor and master degree from plant pathology. Now from land development department. Okay, from Department of National Park, Wildlife and Plant Conservation. Okay, who else? From Marine and Coastal Resources. Dr. Thanuvong Sangthian, my former student. Who else? And we have the author and co-author of these 2 books with us, Professor Ajahn Annong Jansikun here. She is the first author of this book, and Associate Professor Punpilai Suvarnalit, and also co-author of this book. Of course, myself as well. Now, I invite the Dean of the Faculty, Dr. Jongrat Vachalindrat, to deliver the welcome remark and also open the special lecture for today.
Thank you, Dr. Thailand. Good morning. This senior professor of Sakaerat University, Dr. Alexander, Dr. Taiwan, and the head department of forest biology, and all staff and students from Sakaerat University, and our distinguished guests from many departments. As Dr. Taiwan report, actually Dr. Taiwan is one of the CNN of our faculty. CNN reporter. So that's why I think I very surprised because there are many many lecturers and professors come to join today. Even this, I think just only 10 or 20% will join for this seminar. But because of Dr. Taiwan is a senior reporter, as we know already, so many lecturers and students come. For our colleague, Dr. Alexander, so I met you many times. So as the researcher about the microfungi, in my opinion, I think the microbiology is very important for the future trend of our economy, for the global economy. Bio-based economy is very important. So in my target, I would like to promote the bio-based economy. So I call it this eco-economy. How to develop, how to use the organisms to be the eco-economy of the world. Of course, last 50 or 30 years ago, we talked about the petroleum is very important for economy of the world. But now, time to use the microbiology and also some kind of the organism is important thing for develop our economy of the world. So for the microfungi is one of very important to develop as a large scale of economy of the world, and sustainable development also must start from the micro or some organisms. So on behalf of the Faculty of Forestry, we would like to thank you, Dr. Alexander, who come to deliver your report about the macrofungi of the, how to say, Wienerwald. Wiener forest, right?
Wienerwood.
Yes, Wienerwood. Yes. And compared to the Sakaerat, and try to surveying or try to inventory the macrofungi and barcoding is very important. So if we can get the knowledge about how to barcode it, maybe we can use for collecting the data and get the information for the worldwide. So thank you very much again for your kindness to give a talk today. And finally, I would like to declare that our seminar is open. Thank you.
I will not let Dr. Uben start his lecture now. I forgot very important person who also came, who sponsored this book from the office that sponsored this book, Dr. Lakaket from Bido. He gave us, this office gave us the budget to publish this book. From biotech, Bob, one more person, and of course many professors from our Department of Forest Biology. 5, at least 6. Okay. The topic of special seminar today is inventory, barcoding, and distribution patterns of macrofungi in Biosphere Reserve, Wienerwald. I spell Wienerwald, Austria, and Sakaerat. Sakaerat is also Biosphere Reserve. Okay, and I would like to ask Dr. Thamrat Kampajam to tell you the biodata of Dr. Uban. Very brief, because I talk a lot earlier.
Thank you, Dr. Thailand. I'm so pleased to be here and have a chance to interview our guest speaker from Austria, Dr. Alexander Uben. He is an associate researcher at Department of Botany and Biodiversity Research, Faculty of Life Science, University of Vienna. He is a great experience on ectomycorrhizal relationship and macrofungal diversity and ecology. He is also an expert in identification of fungi involved in ectomycorrhizal formation by classic microscopy and also DNA analysis. He also has experience to do research in many countries, including Thailand. Last year, he came visit our faculty to do research with Dr. in Taiwan. We learned a lot how to identify ectomycorrhizal fungi from the root. This is all just a briefly of his experience.
Now you can start. After waiting for more than 10 minutes, please do.
Thank you so much for the warm introduction, and thanks to everybody for coming. And yes, I'm really excited to speak in front of so many outstanding mycologists who will have some relationship. I think the introduction already said a lot about the topic today. I will just shortly speak about the bioreserve in Austria next to Vienna, which was created only in 2005. It was created only in 2005 and covers a total of about 100,000 hectares, of which 5% are core protected forest area. It means that there is no timber extraction anymore and no forest management, except for maybe some biodiversity reasons. And the region is characterized by quite strong geological variation. So there are calcareous areas and areas with siliceous soils, which results in a very high diversity of soil types, which is very important for fungal diversity. The elevation is interestingly very much comparable to Sakaerat Biosphere Reserve. It ranges from about 250 to roughly 800 meters above sea level, so it's in the colline zone. It's not very mountainous. And also it's at the limit of different climatic zones, and part of the area is partially arid, it's sub-arid, while most of the area is more humid. And also this is quite similar to Sakaerat. So I didn't know all this in advance, but when I came to Sakaerat, I was really surprised to see all these similarities. Even though one is in the emerald zone, the Wienerwald is the emerald zone, which means there is cold winter, snow in winter, and the summer is quite similar to here. Maybe the rain is less heavy, but not even. But else the climate in summer is pretty similar. Just the climate in winter is very different. So there is hard winters, but not too hard, like maximum minus 10, minus 20 in winter, which means that all the trees, at least the broadleaf trees are deciduous, not the pine trees, of course. And here you see a map of the forest ecosystems in Austria. So everything which has a color is forest, but the forests are of different naturality. I mean, some forests are pretty much natural in terms of species composition and age composition. And these are marked in dark green or blue. And some forests are more impacted by human forestry and other human manipulations. And these are marked in yellow or even red. So here we are, and this is the area where the Biosphere Reserve is. So we are in an area with mostly moderately strong impact of human activities, with a few patches of quite natural forest. Also, this is somewhat similar to Sakaerat, I would say. And here you can see the area in more detail, and that's the points where I did the recording of the fungi. And this map is, you see here the core protected areas marked in green. This is where, since 2005, no more forest management is applied. And here this is a geological map. And essentially the northwestern part of the area is on siliceous material, so more acidic soils, and the southwestern part is on more calcareous or dolomitic soils, so basic soils. And the plant communities are very different in these two parts. So this, concerning the substrate, it's more different from Sakaerat. And in the region we have a long history of forest use for firewood, timber, charcoal, lime burning, harvest of pine resin for all kinds of products, hunting, mushroom picking, whatever. More recently, strong recreational use. Nevertheless, a few old growth forest lands are preserved from 180 to almost 400 years old, according to the records we have. And it's very close to a metropolitan area. All this Biosphere Reserve is in part belonging to the city of Vienna. So imagine just an area like that next to Bangkok. You can imagine that there is a lot of pressure on this area. And so to evaluate how effective the protection of the area is, an inventory and monitoring system was installed, and it included also macrofungi. Luckily, it was very not many monitoring biodiversity monitoring programs include macrofungi, so we were very happy about that. And of course, we have a strong intuition that forest reserves are very important for fungal biodiversity. But if you look into the literature, there is not much hard evidence for that. I mean, there is evidence mostly from the boreal forest area, where really you have data on that, a lot of data. But for other ecosystems, there is much less evidence that really fungal biodiversity is, or that forest reserves are important for conserving fungal biodiversity. And of course, since it was an integrated project with a lot of data generated, it was also very interesting to participate in that project because it's also possible to analyze the funnel data in a very complex and to compare them with many other data, which we usually don't have as mycologists. We just go to the forest and collect mushrooms, but we don't know what's the age of the forest, and there are many things we don't know usually. And within this project, it was possible to have rich other data to compare. So within about 119 sampling sites, fungi were inventoried, and the sites were selected not by me, because this was considered as objective, but they were considered they were selected by a computer, and then we went according to some complex criteria, and one criterion was to cover as much of the known vegetation diversity. So there are about 28 different forest vegetation types in the region, and one of the aims was to represent these different forest vegetation types in a representative way. And the investigation period was about 2 years, and it's a long period from a personal perspective, but it's a short period for a forest and also for a fungal inventory, I have to say. 2 months here is an even more short period. And I try to visit the sites as well to distribute the search effort as equally as possible on all the sites, to have comparable data, and to visit them 2 to 3, maximum 4 times in different seasons. And well, for identification, we used a combination of classic morphological identification and DNA-based. And about the results, so in this all this inventory, we got about 5,000 GPS reference records. So every single record of a fungus usually has a GPS record with maximum 10 meters error, and about 2,000 specimens were in addition analyzed microscopically. So the most easy things were identified directly in the field, and then about 2,000 specimens were brought home to be analyzed in more detail, and about more than 400 now have ITS sequences attached to them. And more than 700 species of macrofungi were recorded in the sampling sites, and more than 100 species more were recorded just on the way to the sampling sites. So that's logical, because the sampling sites were only of a limited surface and they cannot cover the whole diversity. And of these roughly 800 species, at least 17 species are not yet recorded in the database of fungi of Austria, which means there are new records for Austria. And some additional records for very rare fungi were also made. For example, Glossoporus quercinus is known from a few sites in Europe only, and only from 2 sites in Austria so far. And we found a 3rd site. And very interestingly, we could also rediscover some of species which were described from exactly that area about 100 years ago, and which have never been detected ever since. Like, unfortunately, I don't have pictures of them because they are really very inconspicuous. It's not something very easy to find. That's also the reason why they have not been discovered ever since. It's Thracispora araneosa, Hyphoderma subtestatium, and Myocystitiatum clavoligero. And many collections are still unidentified, and some of them may represent undescribed species or species not yet recorded from Austria. So this is an example of the Austrian database of fungi. And here you see the query for one of the species, Piptoporus gracinus. And it's present in this site here in the southwest of Vienna in an old park. And here in one site in Styria, and we found a third site. These sites are old literature data, but the species have not been re-recorded there for a long time. Okay, this was the basic information about the results, and now I will like to introduce you to some of the species which were new or noteworthy. Since I conducted the research throughout the whole year, even in winter, I went collecting fungi. I found a lot of things like this, without fruiting, classic fruiting bodies with a style and a cap, but rather crust-like fungi, which, however, are very, very diverse. And it's a group of basidiomycetes of it, which is also scattered phylogenetically across the basidiomycetes in general. So they are also phylogenetically extremely diverse, and for that reason are also very interesting from a general systematic perspective. So this is one of the species which was recorded as new for Austria, Dacryopholus castani, and it's not uncommon in southwestern Europe or Western Europe, for example in France, but apparently it is quite, it does not like a very continental climate, and maybe the fact that it is now fruiting in Austria may be linked to the warming of the past years, the global warming which is already happening. It's just a hypothesis, but it may be the case. This is another species which was interestingly recorded only a few years for Austria, first time, Flavia notophagi, and it was, according to our results, one of the most widespread species in the old, but only in the natural forest, in those places, in those stands which have really old age. So it's probably one of the indicator species of old growth natural forest. And this is another interesting species, Tyromyces leonius, and this species may just represent the opposite of Dacryopholus. It likes humid, cool weather, and it's widely distributed in the subarctic zone. And I found it only once in one of the moistest forests, and it may be the case that this species is getting rarer due to climatic change. And this again is one of the more thermophilic species, Climacodon pulcherimus. Probably it's not long time ago, it's about 15 years ago that it was first reported from Europe, from Spain, and it's still rare, considered rare all over Europe. And the genus is quite present in the tropical areas, but it's very uncommon in the temperate areas. This is one of the species which is interestingly known from one side in Austria for a long time, but only from a very limited region, within the area of the Biosphere reserve, and there is no other records from all of Austria. And and it grows only on deeply decayed wood of massive oak trees, which is a rare substrate. And this is the first ectomycorrhizal species. It's associated with oak trees, and there is a lot of taxonomic confusion about this species, and it's a little bit difficult to recognize because there are very similar species associated with beech, with Fagus sylvatica, but this one is exclusively associated with Quercus, and I could find it only in a single site. And well, this is another not very attractive species at first sight, but the spores are very specific, and it's an ascot seed, very ornamented spores. And this is one species which was described only in I think 2004 from a forest in Crimea, Ukraine, and the species was discovered only recently by colleagues from Slovakia and by us in natural forest sites in Austria and in Slovakia. So apparently it has been overlooked for decades. And there is still another example. Now here we'll present an example of how to use the ITS data. I will use one genus as an example. For the genus Anthodiella, we have recorded 5 species, 4 of which are identified, and the 5th species is still uncertain. And the genus Anthodiella is very, very interesting. It's a polypore species, and you can see one of the more bigger fruit bodies look like this. Most species are small, a few centimeters large maximum, and some of the species are just crust-like, so not very conspicuous. But the interesting thing is they don't live on the dead wood proper, but they live on dead polypores or on the wood already decayed by other polypores. So they are very high in the trophic pyramid of the saprotrophic system. And as you may know, that in ecology the top predators are quite important for the indicators for the ecosystem. And I think it's maybe they are somewhat the top decayers. So they use already the other fungi which have already decayed in the wood. And I think it's a good sign that we found so many species of this genus, very interesting genus. And interestingly, one of the species we were not able to identify with certainty, neither with ITS nor with morphologically. And as you can see, this is the one species. This is Anthodiella affine Rohegi, and we found ITS sequences which matched ours 200%, but they came from environmental samples only. They came from air samples. These were just DNA extracted from spores found in the air or samples like that. But we don't have any fruit bodies, no identified species yet. And well, this will be a taxonomic question still to resolve. If there is a valid name available for this species, or if it will be necessary to create a new one. Interestingly, the closest named species is from China, Anthodiella stipitata, but it has a different morphology, and it's unlikely that it's really the same species, and it's not identical in ITS sequence. So this was about just a little example about the use of the ITS data. So when it comes to really making sense of this DNA data, it's a lot of I think when you have the DNA results, the work on the stars, then the systematic work should begin. Concerning the species frequency distribution, as you can see here, most or almost half of the species were found a single time. These are all these species, only one time. And about more than 100 species were found twice, and about another 100 species were found 3 times. So most of the species are really scattered in the environment. And then a few species were very frequent, up to 50% of all plots. You could record a few of these species. And well, when I started the project, I wanted to apply some species distribution modeling with the methods which I still have to learn, I have to say, since we have so many interesting data about the ecological parameters available. But the problem is that for species distribution modeling, you should at least have 20 or 30 records to do some meaningful, to get some meaningful models. And well, so we can apply this species distribution modeling maybe to 20 or 30 species, not more. And this is a very strong limitation. So exactly the rare species, which may be particularly interesting from the point of conservation, are really difficult to model because we have so few data. And I don't know how to get around this problem. Yes. But the only way is to try to collect more data, I think. Concerning the trophic groups, interestingly, these are the 4 classes of frequency. A is the most frequent species, B are medium frequent, and C are the least frequent species, which have only 1 or 2 records. And interestingly, within the most frequent species, almost all of them were lignicolous. The medium frequent species were mostly lignicolous, but other trophic strategies became more important, like ectomycorrhiza, litter decomposing, and so on. And in group C, in group C, the trophic strategies are most diverse. And this is quite easily explained because the lignicolous fungi generally have the most long-lived fruiting bodies, so it's most easy to detect them. While the litter decomposers and ectomycorrhizal species, they only fruit for a short period, so it's much more difficult to detect them. And well, this very much tells us that I think that for that inventory research, classic inventory research, and soil DNA analysis will be very much complementary, because it's really difficult to get the true diversity of ectomycorrhizal fungi from a field study like that. So I think now all people do it with DNA data, and I think I will also try to get a project to do it for these sites with the soil DNA data, because it's hard work to do it with. I mean, I spent 2 years of my lifetime to get this data, and I still have not satisfactory data for the ectomycorrhizal species. While I think I have quite good data for the lignicolous species. So, and one of the questions the managing authorities of the park were most interested in was what's the patterns of species richness. And are the plots and the core areas more species rich? And the answer seems to be yes. So I think they were quite happy with that results. And I have to say that it's really difficult to distribute the search effort equally across sites. But still, mostly those areas which had been protected even before the park area was created. Some of them have a long tradition of protection status. These areas are really the most species rich. So I think that yes, we can show that protected areas are very important for fungal diversity. Okay, to go into more detail, we wanted to know which are the factors which are correlated with the observed species richness. And actually, there was only a few factors which had a significant correlation after correction for multiple comparisons, which is important. If you compare, if you examine many potentially explanatory factors, you have to do some statistic corrections, which will reduce very much the power of your analysis. But some this is called Bonferroni correction, or there are other types. But some of the factors were still significant. And interestingly, the most significant factor was annual wood increment. And this definitely surprised me. It's rather a measure of stand productivity, of forest productivity. And indeed, the most productive sites were also the sites richest in forest, in fungus diversity. Which means that it's important to have also reserves in the not only in the marginal sites of low productivity, but also to have protected sites, areas of which are very productive. And then also, of course, the quantity of woody debris was very important. And of course, some factors like humidity. Probably the dry sites are very rich also, but we just could not observe the true diversity present in these sites, because in the drier sites, well, the fungus growth is so limited and can be observed in so short period only when humidity is sufficient. Maybe a few days only in the year, that it's very difficult to get the best data. So, interestingly, there were negative correlations, especially with light intensity, which means that the open forest sites were less species rich, and also the sites on strong slopes. And I think that this result is rather an artifact of drought, because during these 2 years we did not have optimal precipitation, which means that on the very open sites, fungal fruiting is very much limited by humidity, because the wind and the sun dries the soil very rapidly. So this was one of the analyses about stand age. And interestingly, when we took all the data together, we didn't find the correlation between stand age and number of fungal species. But when we looked more closely at this data, it seems that within this protected area, the oldest forest stands are on the steepest slopes, which means that they are in sites which dry very rapidly and which are difficult to explore. So we found less fungal species in these very steep sites, and and steep and dry sites, and this is why we cannot see an overall correlation. However, there was one group of fungal communities, of forest communities, where we didn't have this correlation between stand age and slope. And interestingly, in this group we found a very strong correlation between the number of fungal species and the age of the forest stand. So we can say that at least for this group of forest stands, stand age is very important. We cannot say it in general, but maybe with soil DNA we will get also a better answer for the more general picture. And another analysis which kind of tries to get all the data together is these are the names which you see here in black. It's the names of the forest communities. So, for example, Euphorbio angulate quercetum is some type of oak forest, which roughly corresponds to, I would say, to the dry dipterocarp carp forest. It's somewhat the equivalent of the dry dipterocarp carp forest in Sakaerat. And, for example, these are forests which are on calcareous areas, and these are forests which are on forest types which are more on siliceous areas. And, for example, the Gallio odoratum fagetum, this is the very moist beech forest. And so there are all different forest types. And interestingly, these data are not based on vegetation data. There are no vegetation data at all in this analysis, except for the labeling of the forest stands. So, but this analysis is based on fungal data. So actually, the fungal communities some kind of help us to find the vegetation units which are based on vegetation data. So there seems to be a strong kind of relation between the fungal communities and the vegetation communities. However, I think we will have to go into more detail in this analysis, since we don't have statistical values yet on that. It's just a pattern. It's not yet there is not yet a statistical test on that, and we will have to investigate it in more detail. And what you see on the blue arrows are the different ecological factors like humidity, nutrient richness, slope, continentality, and so on. And you see that at least in this analysis, some of these factors are strongly correlated. So it's difficult to differentiate these factors from each other. Maybe this graphic represents only 2 dimensions of this kind of analysis. There are additional dimensions in this analysis, and maybe we can get a differentiation of these factors in higher dimensions of the analysis. But this is also still to be explored. So, to sum up the use or usefulness or not of this kind of analysis, the DNA-based identification has a lot of advantages, but there are also limitations, especially the quality and coverage of reference databases. And I think it's also important to do such work to fill this, finally to get more complete reference databases. And interestingly, we could find sporocarps which correspond to sequences which were known from environmental sources so far. So I hope we will also contribute to to filling these gaps in the DNA databases. And are mycological data important for conservation? I would say yes. Mycology is still rarely considered in conservation planning and monitoring. And interestingly, in some plant species, poor forest ecosystems, like for example the beech forest. Beech forest is a very dark forest. There is almost no light on the forest floor and very few plant species. And in this forest, for the botanists, they don't like to go to this forest because they are very boring for them. And so, but the mycologists like to go there. And so probably the microcyanosis are much more informative than the botany for these forest types. And our exploration also showed that macrofungal biodiversity, the knowledge of macrofungal biodiversity is still incomplete, even in one of the best studied regions. I mean, the area of this study has been explored by mycologists for centuries. And we have a lot of data, and still, every time we go to these forests, we may find something not yet recorded. And well, then there is a general question: how can we deliver what conservation agency needs? To my experience, mycology data are largely ignored by conservation planning, and how can we present them in a way that is more considered? And I don't know how much time I still have for the Sakaerat part to describe the project. 10 minutes. Okay, so I will go through it very, very quickly. It's the easy part because it's just images of fungi, no theory, and it's beautiful fungi. So Lycocoprinus sepistipe is one of the most common saprotrophic species. There are one corticoid species, one of the many. I explored only a few of them because it's a lot of work for microscopy. So one of the very many the diversity of Marasmius is extremely high in tropical forest ecosystems, as has been shown quite a few years ago from Thailand, from northern Thailand. Chetocalatus, a fungus for which I have no name yet. Maybe someone knows it. Just a small myxomycete. It's not a true fungus, it's rather a protozoa from a group of protozoa. Something I have no name yet for. It's a basidium. It looks like an ascomycete with this cup-shaped fruity body, but it's a basidiomycete. So it's something close to the genus Zyphella. Well, the genus is well known. Cocaine. I'm not sure if this is Pseudopurpureus striatus, which was recently described from northern Thailand, but maybe. A species of Hymenocete, which is similar to a species I know from Europe, to Rubiginosa, but it's not identical. Some Fabulacea. We have in Europe only as invasive species, as new species, one, but it's very rich here in the tropics. Something which may be quinipelis. I'm not sure about that. Also I'm not sure about the genus for this polypore. Did you get a name for it? No. Some Strobilomys, for sure. For the species, I'm less sure. Philopoletus manipularis. Very beautiful. Something very small. Something like Marasmioid, but not yet sure about that. Here it's easy to recognize the genus and very difficult to find the species correct. Some Entoloma. And Philipsia domingensis. I was happy to find that one. It has some, unfortunately the image is not good enough, but the spore, you should see some stripes on the spores. The surface of the spores has a kind of stripes. And, well, Thailand is famous for cordyceps research, and at least one species on a small bug we could find. Some xerocomas, and this is one species which I dared to identify by the substrate because it was growing on the dung, the feces of wild boar. So I think it's the Cyathus stercoreus. Some very beautiful species, easy to recognize, Trobia infundibulliformis. And also a very colorful species. It's a Lexinum. It's looking quite dull, but when you cut it, it turns orange-red. Lexinum. I think it's Ectospurius. And one species which I have not found in the book yet, it's Ececalciferus. It's very small. It's less than a centimeter large, and it's a bolete. It's the smallest bolete I have ever seen. And this is one of the hugest amanitas I have ever seen. It was like that. you see, this is 15 centimeters down here. So the cap was about 20 centimeter diameter. It's close to Echinocephala, but not identical. Some genus which is very frequent in the temperate and boreal zones, Cortinarius. I think it's less frequent in the tropics. This is maybe some of the typically northern genera, it's Cortinarius, but one species, the only species which I could find. And on the other hand, Tylopilus is we have only one species of the genus in Europe, in whole Europe. And there was one stand with Castanopsis trees, and in this stand alone, with around 3 or 4 Castanopsis trees, there were 3 different species of Tylopilus. And the second species, and I have not a good picture of the third, some beautiful Amanita. Of course, Astreus. This one we didn't see that in Sakaerat. This was from Bantak forest, petrified forest park. I just want to have a picture of Astreus. This is some Crepidotus, maybe close to Crepidotus variabilis, but I'm not yet sure about that. Gymnopus is also very diverse. Former Columnia. And to my surprise, also Agaricus is extremely diverse in Sakaerat. And this is one from the section Xanthodermatae. I don't know the species name as yet, but I think the diversity of Agaricus is huge in Thailand, to my impression. Also, there is a big diversity of Xalaria species, of course, in the tropics. And since I experienced only the early season, I didn't see the mature fruiting body yet. But I saw a lot of different anamorphs, which usually occur early in the season. And I think that this is some of the anamorphs of Xalaria. And this is another type, or maybe not the genus, but from the family. Some beautiful Lepiota, also very diverse. And some mycorrhizal species. All these species were saprotrophic. And this is again one mycorrhizal one, Telephora species. And again a saprotrophic one, a big fairy ring of chlorophyllum. Chlorophyllum, I think, molybditis, which is unfortunately toxic. So, and interestingly, all the Thai people know that this one is toxic. I think because it's so I mean, it's so big, it looks so delicious. So I think someone has tried it. And ever since, everybody knows it's toxic. Okay, this is, well, also very colorful mushroom, Agaricus trisulfuratus. It's a young fruit, but it will form a true cap later on. And this is a genus, I'm not very sure about that, but it may be Hydropus, which is a rather rare genus. And yeah, it was strange because it lacked the usually they have small scales on the cap, so I'm not quite sure, but all the other characters fit for the German, so I don't know. The bolete with the wrinkled cap surface, and some very beautiful lepiota from the dry dipterocarp, quite big, and Flavorbos potentosus, which I think it is not so yellow as it should be, but when it dried it became very yellow. Polyporus, Leptocephalus are very. This is a very beautiful bolete. We found it only once. Also another genus which is rare, Hohenbuehe. Maybe the species is not yet recorded from Thailand. Entoloma, another Entoloma species. Very colorful blue. This is an error. This is not Entoloma, of course. This is Amanita caesarea. This is from the dry dipterocarp forest. And it was fascinating because I found it only, I came back yesterday from Sakaerat to Bangkok, and I found it only the day before yesterday. So they almost escaped my my research. And it was a way I had walked 10 times before, and this one day there were at least 3 or 4 places with the Amanita caesarea in the dry dipterocarp forest. And it's a species which is heavily sought after by mushroom pickers, but it's close enough to the research station that the mushroom pickers do not go there. Another lepiotes. And that's the last 2 minutes. Dr. Yamada from Japan introduced me to the Macrothermes and to the termite fungi and to the termite nests. And this is a nest of Macrothermes carbonarius and the fungus garden. Here you see the soldier, and because now there are not, usually there should be the workers, but now since we disturbed the nest, the workers are away and just the soldiers are there for defense. And these small white things, these are the bulbs produced by the fungus, which the termites harvest and use for food. And this is another termite nest. It's in the bowl of a big termite mound. It's Ancistothermes pakistanicus. It cultivates another species of termitomyces. It also, the nest smells differently than the other one. You can even tell it from this one. And you see here Dr. Yamada at work. This is a colleague, Alison Harrington, who was very helpful during all the collecting. And this is a termitomyces species, and you see the root and the stem of the species linked to the abundant fungus garden. Once the fruit bodies grow, the termite nest is abundant already. So thanks a lot for your attention, and thanks to all the people who supported me.
Thank you, Alexander. I will summarize your talk a little bit. Hope I can do it well. From the 119 sites that he worked in Wienerwald, Austria, he found 725 species of mushroom. and also 103 species outside the sites or outside the plots. At least 17 species have not been reported in Austria. That means it may be new species.
No, these are new records.
New records.
These are new records only, no new species.
species.
But some, I think maybe some 4 or 5 additional are new species, but I'm not sure about it.
Okay. And he also gave us some examples of workings on DNA using ITS lesion. It's too low of the right. He gave us examples of working on DNA and at least 100 species found very frequently, and most of the species found only one time. That's what I got from his talk. I may not be right. The last species must be very important in terms of conservation. And for modeling, ecologists would very like to do how to tell, if you found it, in what condition you will find other living species. So for the modeling, the mushroom that could be used should be the ones that can be found frequently. Okay. Next. In his research, he also divided the mushroom into different groups. For example, the lignicolous, the ectomycorrhizal mushroom, the decomposing mushroom, or fungicolous. That means growing on other fungi or eating fungi.
Yeah.
Okay, and some parasitic group. Now he tried to explain about the ecological characteristics of the mushroom that he found. The plots in the core areas, or it means in the good protected areas, have more species richness. Okay, and factors correlated with species richness or in Thai ปัจจัยที่มีผลต่อความมากมายของเห็ดเนี่ยนะคะ The factors that correlated with species richness is the annual wood increment. That means in the forest site that have good growth, there will be more mushrooms. So it's very important to have very good conservation of the forest. This is what you have to think about. And the place where there are woody debris, a lot of woody debris will have more mushrooms. And also on the moist sites, negative correlation with this is ที่ต้องไปเชิญนะคะ The negative correlation with the quantity of mushrooms are light and strong slope. That means high slope you found fewer mushrooms. And there are no correlation with stand age. And he also said that in the older side, they are on the steep slope. That means slopes are important for the quantity of mushroom. Now we'll stop very soon. He also did the conical correspondence analysis or CCA analysis, which is very interesting. He found that mushroom species had some correlation with the types of the forest. Different forest types have different numbers and species of mushroom. And also there are some correlations with slope, humidity, light intensity, and tree growth. Benefit and limitation of DNA-based identification depends on many factors, but one is that quality of DNA database. In terms of conservation, there are still very few data in conservation and planning and monitoring of the mushroom diversity. Last page. Gone. One more. I think this is very important one. Gone. Okay. At least people or students from microbiology will get some idea that when we did the mushroom diversity in the forest, we do not pay attention only in the species richness. We have to, species richness mean the numbers of species and how many of them. But we have to work more on ecological characteristics. That's why he showed you the CCA analysis. Okay, and the final end of his talk, he showed us the species of mushroom in Sakaerat. He worked on try to pick up mushroom and walk in the Sakaerat area for many weeks, but most of the time he found lots of mushroom only 2 weeks before he came back to Bangkok. The earlier time it was very dry and he found only the polypores that left over from last year maybe.
Yeah, and there was one site in a very steep valley, which is humid even in the dry periods.
Okay.
This was luckily Mr. Taksin supported us. He sent us and gave us a driver to go to the site, which is difficult to get, to reach. And so we could explore it for 1 or 2 times.
Yeah, before I have questions from the audience, I would like you to say some words about the diversity of mushroom in Sakaerat in
general.
In general, so that the young researcher would like to work in Sakaerat.
Well, I cannot put it into relationship with other sites. I can just relate it to my experience in Austria. And I would say that some of the genera are extremely species rich, like Lepioda. I have seen mostly the saprotrophic genera, because I think the season for the mycorrhizal fungi would only start now. I think it will take another month to study the mycorrhizal fungi, which probably will come up now after the rain is. Because when the rain comes, the first fungi to fruit are the saprotrophic ones. And only later the ectomycorrhizal will follow. And the genus which I have seen now, it was Amanita. It's just started. And there were already quite a few species present in the dry dipterocarp forest. And I think also from an ecological perspective, from my think this was the most interesting thing, to see a forest which burns almost every year and still is very productive and rich in fungal diversity. In Europe, we don't have forest. I mean, at least in Central Europe, we don't have forests which burn regularly. I think naturally some forests would, but this is completely suppressed by management. They don't want it. And I think that the ecology of some forests in Europe cannot be understood without the burning. And for me, this was, I think it's one of the most exciting aspects of these sites, to see all these adaptations to how the plants and the fungi can live with this fire and all these things. It's maybe like what they has maybe the same function like frost and winter in
Okay, thank you. Now, questions from audience. Please, Dr. Jongrak, you have any questions? Please.
Actually, I'm not a mushroom man. I'm the forester. Anyhow, you mentioned about the relationship between the mushroom richness and annual increment, annual wood increment. So my question is, actually, in like a vegetable forest, it actually no growth. Maybe anyhow the biomass is very high, but no growth, no increment. That means very low mushroom richness, right?
No, okay. I said, I mean, you have no net increment because,
yes.
But my measure for increment was the size of the ring of the width of the tree rings. I don't know if you can do this probably in the seasonal forest here. You can do it. You can count the years and how to explain. It's just the ring with the size of the ring of the annual ring, and of course in the natural forest this slows down a little bit.
Yes.
And the highest annual increment is if the stand is maybe 20-30 years, or I don't know when the productivity is highest. But I think it's more site specific than management specific. I mean, even you have also trees of 20-30 years old in the old growth forest, because you have natural regeneration. And so I calculated it for the trees, which not for the very big. I used only the trees with a certain diameter to calculate it, to have a base, to have the same base for comparison. So I used it as a stand characteristic of potential stand productivity, and not as and I used the stand age as an independent factor. So I tried to filter, to separate these two, but it's true it's not easy since both are linked.
Other questions, please?
I am interested in your introduction about the genus Anthodyella.
Anthodyella.
Why you give interest in this species, and I cannot remember well that what difference between the Anthodia and Anthodyella.
Anthrodia produces a brown rot. Anthrodia produces a brown rot, and which means the wood decayed by Anthrodia has a typical patterns of a brown rot, and wood decayed by Anthodyella has typically white rot. But in fact, I think that it's not the wood which is typically so much attacked by Anthodyella, but it's the white rot fungi which are attacked by Anthodyella.
And whether it's useful to as the medicine or something like that.
That's a very important question.
Do you have any effect on that?
Yeah, I have no data on that for this study. I know that I think biotech explores a lot the properties of fungi.
maybe answer from Bob.
So biotech working on a lot about the bioactive compound from the mushrooms, but not from this genus, but for beside of the genus such as the Ganoderma and Styrium Austria, or other else that they produce a lot of compound, new compound, which is against the bacteria and antivirus and anti-TB also. Yeah, that's for an example. In fact, that they're more than this.
No work on Anthodiella or Anthodiella yet.
No, we never have that.
Well, that open to the new topic of thesis.
Exactly.
Any questions from students? Or any questions from anybody? Yes, please.
Thank you for the presentation. I have one question.
Closer to the mic.
I'm sorry. I have one technical question. From a single building sequence, which data database you use?
Well, I used mostly the mass blaster of Unite because it including the you have two options. You can include the gene bank data or not, and including the gene bank data. So you can access both databases with a single analysis, which is the fastest way to deal with a lot of data.
You are from Land Development Department, right? French. This room gather all the microbiologists, mycologists, and foresters, plant taxonomists, many people. Ajahn Sarawut, you have any questions?
No, thanks, I don't have any.
Titima.
May I ask you about the primer that you're using? Regarding of your presentation, you're talking about the working on the ITS, right?
Yeah.
So how about other primer that you use?
Well, actually, I have worked with different primer combinations. I did not have the same primer all throughout the study. Normally, I tried in the first run to get a big piece of the DNA, including a large part of the 28S or LSU, using the primers ITS1F or ITS1 and LR5. So you have about the whole ITS plus 900 bases of the LSU. But it's almost 1.5, 1.7 kb, and you don't get this fragment for all the species. So then sometimes I had to do only the ITS. And the LSU is more useful when you have in certain groups of fungi where you don't have the DNA data yet. And some people worked more with LSU with ITS. And so it's good to have both.
So do you think, in your opinion, there are any primers that useful for doing a barcoding besides of the ITX or the LSU?
No, there are other regions which are needed, but this is fauna specific. When you have in penicillium, you need many different, for example, many additional primers. But here, it was a project which was not focused on a specific group of fungi. So I used the most universal ones. And if you focus on a specific group of fungi, you have to use, of course, more specific primers.
Thank you.
That's great.
One last question.
May I have one more question?
One more and then one last.
Okay. You say about some species that you found only once after 100 years ago. So it's in your opinion, what's the answer of why 100 years ago never been found and why now?
I know it for a few. In our database, it's also in part it's taxonomic opinion. sometimes the species are lumped together, and sometimes they are too much. Sometimes they are split up, and they were described by people about 100 years ago, who rather split the species up. Ever since, there has been a tendency to lump several species together, and so they just went undetected. They were always labeled. This is true for one or two species, and only now with the ITS data we see that this old opinion from about 100 years is true. So it's really different species, and we should use the name of the people who described them 100 years ago. But in one species, it's very characteristic. It's just, I think it's just so inconspicuous. I mean, you have, if you look at it under the microscope, it has crystals like a butterfly. You cannot misidentify the species when you look at it at the microscope. I think just no one collected it. It's fine.
Someone collected and could not identify, then throw it away. That happens very often. Okay. นิสิตอยากถามอะไรไหมคะถามเลยอาจารย์แปลให้อาจารย์แปลเผื่อรู้มีไหมคะคุณดร.รัชเขตมีไหมไม่มีอาจารย์จงรักโอเค
Of course, I'm the forester again, and I mentioned about the eco-economy. So I interested about it. Why the commercial mushroom is just we found a lot in the like a dry forest or dry zone or open forest. Why not in the moist evergreen forest? Can you answer?
Well, maybe there is a socio-cultural. I thought about that question too, and I think there is a socio-cultural reason to that in Thailand. It's my opinion, but I would be happy to know your opinion about my explanation to that. But as far as I have seen, mushroom picking is done by women only, or mostly in these forests. And as there are many mycologists who are women.
You mean women don't like to go deep in the forest?
And I don't know, but I thought that they went in a big, or maybe in a small group of 5 to 6, and they like to. I think the open forest is much more. Even though there are many dangerous snakes and so on, but it's more. You feel more comfortable than in a very thick forest. And we found when we went to the thick forest, we found a lot of edible bolides. There is no reason not to go to the evergreen forest to pick mushrooms. But I think the open forest is just the more. It corresponds more to the ideal human landscape. It's like a park, and I think it's. It's not so easy with the bamboo grass, but if you know the places, but it's just a more you see far away.
I have also some answer about this. In the dry dipterocarp forest, especially in Sakaerat, there are dipterocarp trees that support a lot of ectomycorrhizal mushrooms, and some of those are edible, and the villagers really like it, like Amanita princess or เห็ดละโงกขาว and also some Lassula เห็ดแดงเห็ดน้ำแป้งเห็ดตะไคร้นะคะที่ขายอยู่บนท้องถนน the Lassula species, and for the economics mushrooms that grown by mushroom students mind and also professors. But anyway, the time is over for more than 20 minutes now. Before you leave the building. If you haven't got the curly puff, you are welcome to get it back. One only for everyone. And may I invite Ajahn Anong to give some souvenir to Dr. Uben. It's the mark that have my name on it. Please give it. ให้อาจารย์หนึ่งอย่างเดียวค่ะเดี๋ยวอาจารย์คะใครมีถ่ายรูปด้วยอาจารย์ถ่ายรูปเดี๋ยวอาจารย์จงรักมีถ้วยอีกใบหนึ่งให้เอาโซย่าหนึ่งสิคริสค่ะ Our soya has came last year as well. When Martin is 1 year old, and now Martin is 2 years old and have another baby. Our soya likes Thailand very much. Okay. And Dr. Jongrak asked whether mushroom scientists have to be women or female. No, there's no limitation. And like Alexander's and also his friends in Austria, they are all men, gentlemen. ชอบล้อยายถิ่นอย่าง Okay, thank you very much. I hope that we will have some other special topics to invite you to come to our faculty again. Thank you.